Okay, question about plants, photosynthesis. Here you've got a section through a leaf. It's a cross section through the leaf, and the palisade layer is labeled. So you need to know facts that palisade layer is where most photosynthesis happens in plants. And the first question is asking you to say how it's adapted. What's special about it? Now the dots on this diagram are showing the chloroplasts, and you can see that there's more of them in the palisade layer. And so the adaptation of the palisade cells is that they contain a lot of chloroplasts in order to do photosynthesis. You could also mention that the palisade layer is at the top of the cell, so it's going to get more light. And again, that is an adaptation to help it do as much photosynthesis as possible. Part B. Gases pass into and out of the leaf through small pores in the surface of the leaf. They're labeled on the diagram X, and they are stomata. Don't get confused here. The hole itself is called a stomata, and the cells either side of it, which can open and close those holes, they are the guard cells. The guard cells are guarding the stomata. We've got another calculation here, this time calculating the real length of a cell. Get your highlighter out, highlight the numbers in part C. It says the cell image measured 28 millimeters in length when viewed at a magnification of times 400. We need to do image, so 28 divided by 400, and that is going to give you an answer of 0.07 millimeters. The second part of this calculation asks you to convert your answer into micrometers. So we've got an answer of 0.07, so we need to do something with that. We're told that there's 1000 micrometers in 1 millimeter. And so we're going to times our answer by 1000. The right answer here will be 70 micrometers. Part D, carbon dioxide can move into and out of cells. How does carbon dioxide move? We know that it's going to move from where there's more of it to where there's less of it. Carbon dioxide is going to move by diffusion. There's some diagrams trying to show numbers of molecules here. Okay, so if we're looking for the fastest rate, the main factor that affects how fast diffusion happens is the difference in concentration. So you're looking for the biggest difference between inside the cell and outside the cell. Cell A is going to be where diffusion happens fastest. And our reason, and you must get the correct cell to get this mark, the reason is that there's a bigger difference in concentration of CO2 levels from inside the cell compared to outside the cell. Or you could say in cell A, there is a steeper diffusion gradient. Second part of this, we've got a diagram. It's the sort of setup you would have in your required practical to investigate photosynthesis. Read the information carefully at the top. It says a student investigated the effect of different colors of light on the rate of photosynthesis. So whatever they're investigating is it's like the title of your practical. Yeah. How do different colors of light affect the rate of photosynthesis? The student placed the apparatus in blue light, then in green light, and then in red light. The student measured the rate of photosynthesis in each color of light. So the first question is asking you, what do you need to measure in order to calculate the rate of photosynthesis? What would you do if you were working out here? The bubbles are being made by the pondweed. They're going to be oxygen. And the rate of photosynthesis would be how many bubbles are produced in a certain amount of time. So the two measurements are the number of bubbles and the amount of time. give two variables the student should keep the same. So we only want to change the color of light. That's our independent variable. Anything that could affect photosynthesis needs to be kept the same. So our big three are temperature, light intensity, and the concentration of carbon dioxide. So those three factors are the big things that affect photosynthesis. So if I was doing this experiment and changing the light, I need to keep the temperature the same. I need to keep the light intensity the same. So remember, we'd probably do that by keeping the distance of the lamp from the pondweed at a fixed position. And I need to keep the amount of carbon dioxide the same in the water. I might add some sodium hydrogen carbonate to the water to make sure there's always plenty of carbon dioxide so the pondweed can do photosynthesis well. You're being asked to complete a graph here. Notice that it's telling you exactly what to do. If I was doing this question, I would be crossing off the things on those lists, those 4 bullet points there as I go. Yeah, I need to label the y-axis, so that's the vertical axis. It's going to be whatever is on the top of the right-hand column of the results table. So you can see color of light on X. We want rate of photosynthesis in arbitrary units on the y-axis. We need a scale. So looking at the numbers there, the biggest number we've got is 9. The smallest is 1. There's a 0 already on my scale. I'm going to count the squares. 2, 4, 6, 8, 10. I'm going to go up with each 1 centimeter square as 1. And that means I'm going to go all the way up from 0 to 10, and I will be able to fit on my points and use most of the paper. Plot the data. Think to yourself here, the color of light is a label. So the correct type of graph here is a bar chart. It's telling you to plot a bar chart. We need to fit 3 bars on, 1 for blue, 1 for green, 1 for red. Finally, label the bars at the bottom and make sure you've got the right label on the right bar. The final part of this question on photosynthesis asks you to pick out which color you would use to grow plants in a greenhouse. And you can see from the numbers here that although red and blue are almost the same, blue is slightly better. So blue causes a faster rate of photosynthesis. If you are lighting a greenhouse with artificial light, blue light would cause your plants to do photosynthesis quicker and grow faster. Green light would be hopeless. Look at the number 1 on the table.